Thank you very much, Matteo, and the other members of SysMod COSI for the invitation to speak here. I'm delighted to present to you today work that's been ongoing for a number of years. It's come to fruition at the moment. So any of you can download this PDF of this presentation via this QR code and also at the end of the presentation. So I want to give you the outline is basically kind of the problem statement, like how we do kinetic modeling at genome scale, and then describe to you the progress we made in the early years, the kind of mid-era, where we kind of changed track or sort of went ahead and just computed with what we had, even though we wanted more in terms of what constraints we could integrate. And now the more recent developments where we've introduced new methods and numerical results that seem to indicate we're in a good position. I presented some of this work already in Stellenbosch and in San Diego, and I was in the middle of writing up this work, and I came across a hole in it, and I had a panic about it. And this cartoon scare at bedtime from Podge and Raj came to my mind. So yeah, relief in the end, and the last couple of months I was able to demonstrate global convergence, and but that means the paper's not yet finished, so maybe by the autumn it'll be out. Then I'll conclude with some ideas about future work. So essentially the situation is that there's like a paradox in genome scale modeling, where most genome scale modeling methods only explicitly model reaction rate and not molecular species abundance. So there has been like efforts towards genome scale kinetic modeling, and but then none of those are really scalable. Like, so they typically maybe 300-400 dimensions, or maybe even up to 1,000. But the methods behind it are not scalable. They're not globally convergent to an optimal solution, like in the way, let's say, flux balance analysis is. The problem with this is that the experimental measurements, they all measure abundance of things, but we're in the genome scale modeling, we're mostly modeling reaction rate, and so you kind of have this problem of validating your predictions, because one of the measurements are of abundance, and the predictions are mainly of fluxes. So you can kind of look at this in two ways, and one way is, maybe let's say from like across the pond, you might look at it and say, like, how do you constrain genome scale models with reaction kinetics? But in Europe, you might look at that same problem and say, how do you extend reaction kinetics to genome scale? So this project or program started off during my PhD when I went to UCSD and visited Bernard Paulson's lab, and I just tried to pick what I thought was the most important problem in the field at the time. Totally naive and in a way, because it was almost career suicide to have done so. But anyway, I started working on this problem back then. So just what we're talking about here. So I guess many of you are familiar with this, but essentially we're talking about reaction kinetics, and especially systems of reaction. So this is a very simple reaction here. You'll see multiple times in this presentation the exponential and logarithmic terms, and those are meant component-wise, and these are some basic logarithmic and exponential identities. And so they are important because they can be used to formulate reaction kinetics instead of formulated in this form here, which you will often see, like this power term. You can formulate it like this in terms of exponentials and logarithms. It's the same thing. It's just reformulated to use more matrix vector notation. So personally, I find this is absolutely instrumental for understanding what's going on, because this, it might seem a little more clunky at first, but when you start using it over time, it really brings out the matrix vector aspect of it, and it's really useful for computing all sorts of things like derivatives and things using matrix calculus. And so it's reformulation, but it's instrumental. So at the very early stage, the first paper I ever published was basically showing that you could do integrated stoichiometric thermodynamic and kinetic modeling of steady state metabolism if you could find solutions that were the intersection of a polyhedral convex set, which is the set in navy there, and a polyhedral convex set that's transformed into logarithmic scale. And so that's the set in red there. So the intersection between those two, like if you could find solutions at that intersection, you would be solving this problem. And so what I realized with that paper was that basically it's far easier to put all the equations together than it is to solve them. And so this is kind of reminiscent of the maxim of Dirac, although this is physics and it's a different arena. But you know, they were referring back then, 1929, the pre-computer era, to solving these on paper, you know. But it's still true today for even if you're solving them numerically. So a large part of the early work, like I was very naive. I really didn't know how to approach this problem. I kind of learned what I needed to learn as I went along. And so I spent months trying to find solutions to systems equations. Eventually, I realized that there was sometimes no solution. And so this result here is kind of given an idea, when does a non-equilibrium kinetic steady state exist? And so that it exists when you connect all the inputs of a biochemical network back towards the outputs. And so this is reminiscent of how you see always in electrical networks display. They always close this loop, but then they have this battery term. But we tend to show metabolism with inputs and outputs, and they're not connected to each other. But of course, in life, or else the mass is not connected to each other. But you can, in the computers, create fake reactions that connect the mass going out one side to the mass going in the other side, and at steady state they'll be balanced to each other. And so then the trick then is to you add let's say a driving force, like a battery, essentially, one for each conserved moiety, and that if you do that, then you're guaranteed that a kinetic steady state will exist. Subsequently, I was introduced to convex optimization, and here the idea is that you minimize a nonlinear but convex function that has one unique global minimum over a convex set. So the convex set is in red, and the convex function is illustrated by these, let's say, contour plots here. And I was introduced to that technique by a fellow called Michael Saunders from Stanford, and at the time I thought it was hocus pocus, but I could see it was solving, at least for steady state. It could solve for a non-equilibrium thermodynamic steady state, but not kinetics on its own. And I could see it work numerically, and I didn't understand why. So then I delved right into the convex optimization literature. So at the time, it also didn't include, that formulation didn't allow us to include bounds and reaction rates. So it kind of impeded. It was a nice theoretical result, but it impeded its ability to be applied to real systems where you do have bounds. But 5 more years or whatever working in Iceland, I was able to show numerically that a sequence of convex optimization problems where the optimal solution to the first problem changes the parameter to the next, and you keep iteration this for, let's say, 25 iterations, you can converge to a non-equilibrium kinetic steady state. So it's irrelevant really here what those terms mean, but that plot is probably maybe 10 years old, or maybe 7 or 8 years old at least. The plan was probably 10 years old. Anyway, that 5 years in Iceland, I realized that basically you have 3 different constraints, right? You have the non-equilibrium steady state constraint, you have the thermodynamic constraints, and you kinetic constraints. And so with any 2 of those constraints, you could solve them with a single convex optimization problem. But if you wanted to solve for all 3 via a sequence, then there's 3 different options, because you can choose any 2 first, and then you iterate to the 3rd. So that was, really interesting and really fascinating work. But unfortunately, the mathematical results from the literature at the time, it couldn't prove convergence of those sequences, even though I could see it numerically working on genome scale. We couldn't prove it would converge because the problem was the problem is at that time we thought it was monotone, and it is monotone in that dimension, and I'll explain what I mean there. So a monotone function here is this one on the left, and on the right you have a non-monotone function. So those concepts generalize to higher dimensions. In fact, we were able to demonstrate subsequently after in Luxembourg that they're definitely not monotone. It's definitely not monotone function, and so we found it's a new form of generalized monotone function. So there's a whole family of non-monotone but have some properties of monotonicity, and we named it Duplo monotone. And at the time was because my son was playing with Lego and he had this Duplo Lego, and so that's how the term came along. But unfortunately, while we could show they were not monotone, we couldn't show that they were Duplo monotone. We could show in small examples, and so then that was also another problem, because if you can't show your problem is indeed Duplo monotone, even though I could see it numerically, then you can't prove that it's really globally convergent. So this algorithm is globally convergent if you assume the system is Duplo monotone. So that there was, let's say, started the project 2015, and this is 2011, and I was really at risk of having to exit academia because I'd spent so long working on something that was, only productive in my mind. I could see the progress in myself, but out to the outside world, there wasn't much, and we had reached an impasse at that stage. But through that time, what really started to increasingly do, and this culminated in this paper here recently, is to try and examine what's the unique properties of a stoichiometric matrix. So how many of you have ever done an analysis involving a stoichiometric matrix? Hands up. Yeah. So keep your hands up. So how many of you know for a fact that the algorithm you used exploited the fact that that was indeed a stoichiometric matrix as opposed to an arbitrary rectangular matrix? Yeah. So don't see any hands up, but if you did have your hand up, you can talk to me after. So in fact, stoichiometric matrices are a unique, not unique, they're a special class of matrices that correspond to a special type of hypergraph, right? So they're a special type of hypergraph. Essentially, it comes from the chemistry. I can't go into the details here, but this process of trying to identify more and more restrictive sets that satisfy different mathematical, let's say, constraints allowed me to eliminate matrices that were causing problems numerically. Yeah, because we had a family of different models I was always testing on, but I needed a way to try and say, okay, at least these set of models of different dimensions, they satisfy these set of mathematical properties on the way in, because I didn't know. I was trying to see if we could prove convergence would be related to the structure of the matrix. So we did a lot of work also then on trying to generalize the thermodynamic feasibility results to include bounds. And so then we had the problem to identify models that had still the bounds, upper and lower bounds, but still admitted a thermodynamically feasible solution. And that culminated in this thermokernel algorithm, which allows you to extract a thermodynamically feasible metabolic model. And the motivation behind that is that a kinetically feasible steady state is a kind of a subset of thermodynamically feasible states. This culminated in this axiomics to model pipeline. It does many other things than that, but essentially you take a generic model and via any sequence of very many different steps, you end up with a cell type specific model. We apply this to, in this case, it was dopaminergic neuronal metabolism, and we were able to show that the models have better predictive capacities than alternative methods. So this paper is hoped to appear in computational communications biology shortly, but a huge amount of effort went into it. And I don't think most of the time you don't see people trying to do quantitative predictions with constraint-based models, except of the objective. So what we're doing here is trying to predict the uptake and secretion rates of a perturbed dopaminergic neuronal metabolic dopaminergic neuronal cell culture, like iPSC derived cell culture. So that you know, we get it qualitatively better than semi-quantitatively, but still we were happy to get a Spearman row 0.48, and because I think you just don't see people doing that. So that allows me to make a bigger point here. So we submitted that paper to Molecular Systems Biology and was rejected without any review. In my opinion, that journal has been taken over by people that don't do any modeling at all. And so they either change the name of the journal, they're going to have to change the editorial board. So constraint-based modeling of macrophages then was a follow-up project, and we applied that same pipeline to try and predict effects in Gaucher metabolism, and you can talk to Yan-Jun more about this at her poster. But essentially what we see from the comparison that was available in the literature is that this approach does correctly predict a lot of the reported metabolite changes and changes in metabolism associated with Gaucher's disease. But there's many predictions we just don't have any information for, and we have an ongoing project now to try and validate more of these predictions. So the thermodynamically feasible non-equilibrium steady state we had, and we have, let's say, predictions with that, and we think it's better than non-thermodynamic feasible steady states. But I still always wanted the kinetics in. That was the original motivation. So we had this problem that the function, if it's monotone, you can solve it, but if it's not monotone, we can't solve it reliably. So you're familiar, many of you, with linear optimization. So in this case, you have a polyhedral convex set and a linear objective. You find an optimal solution. But we should also consider other types of sets to optimize over. So for example, both sets on the left are convex. The one on the right is not convex, but of the sets on the left, the set on the far left is nonlinear, but convex. Right? So the big dividing line here is not between the linear and the nonlinear, it's between the convex and the non-convex. So every function, this is not shaded so well, but above the epigraph of a function is the part of a function that, let's say, if you were to fill with water, the top, that's what would fill up, is the epigraph. The hypograph then is the piece below a function. So the epigraph of a convex function is itself a convex set. So this set here in green is the epigraph of f of x, restricted within the bounds, upper and lower bounds on x. So from every convex function, there exists also a convex set via its epigraph. So, for example, consider this what's called convex cone, right? This is the Euclidean cone, so it's non-linear because it's shaped like a traffic cone, essentially, right? And this is called the Euclidean cone, and these are convex sets, like so the interior of that, any 2 points in the interior you can see, so you can optimize over these. So conic optimization is then the study of optimization over cones. So the simplest cone, right? Many of you who are doing constraint-based modeling already been operating over a cone, but you didn't know it is the positive orthant, right? That's the simplest cone. So anytime you put a non-negative constraint, you didn't know it was a cone maybe, but it is can be considered a cone. But here we have a non-linear cone, and we can optimize over it in a similar way. So the particular cone I really want to introduce to you now is one apparently was introduced in the early 1990s. It's called the exponential cone. So the exponential cone is three-dimensional. It's defined by this relationship here. So x1 greater than x2 times exponential of x3 divided by x2, right? So actually, we're only interested in a slice of that cone where x2 is equal to 1, right? So when x2 is equal to 1, this cone is the same as this expression here, x1 greater than equal to exponential x3. So that defines the epigraph of an exponential function. So this set here, right, above the exponential function is the set then that we're interested to optimize over. So perhaps you can appreciate that, if you can hit the boundary of that set, then you'll have a nonlinear relationship between x1 and x3, which is basically x1 equal to exponential x3. So the boundary of this set, the exponential face, is defined by x1 equal to exponential x3, right? And the interior of the set is x1 greater than exponential x3, yeah? So the exponential cone is like a relaxation in a way of the exponential function when you consider x2 equals 1. So then this relates very nicely to many equations that exist in reaction kinetics, thermodynamics, all over the place. Because here we have let's say, for example, the forward rate of the reaction is the exponential of this logarithmic term for the kinetic parameter times the forward stoichiometric matrix and times logarithmic concentration. So what you can do there is, and then you just replace you change your variables from linear scale to logarithmic scale, and you get an expression like this. So v forward greater than the exponential of their forward stoichiometric coefficient times logarithmic concentration plus logarithmic kinetic parameter. And that constraint can then be represented as an exponential cone constraint here. So this is the one for the forward reaction, and this is the ones for each one of the reverse reactions. So each reaction has its own exponential cone, right? Each forward reaction and each reverse reaction, each one has its own exponential cone. So then you can use linear optimization to point you then towards the boundary of that cone, and then when you hit the boundary of the cone, you get the nonlinear relationship you're looking for. So you can go absolutely wild with this. So because the intersection of every convex set is itself convex, and there's a increasing family of cones that you can optimize efficiently after. So what I was able to do then is include terms to optimize the over fluxes and concentrations, maximize the entropy of fluxes and concentrations, minimize quadratic penalties from any given mean plus or minus standard deviation, like a quadratic penalty from deviation for that, include thermodynamically feasible kinetic parameter constraints, moiety conservation. The potential is huge here, right? Because provided you're using a well-behaved cone, you can do this at scale. So how does it numerically? How does it compare? So this is a genome scale metabolic model, and it's about two and a half seconds or so to solve. So it compares favorably with linear optimization. Essentially, the kind of procedure is like here. So you have your polyhedral convex constraints that I mentioned earlier. So anything that can be represented in linear form, and then you put your nonlinear ones, the elementary reaction kinetics, you represent with this exponential cone and the optimization of other objectives that you want to choose. So the intersection of those set then defines another convex set. Part of it is polyhedral and part of it is nonlinear. And then you aim for the boundary where you want to say the reaction kinetic constraints to be satisfied. So of course, the stoichiometric models are based on overall reactions. And so I was able to develop a formulation that incorporated convenience kinetics. And so now the parameters and the variables in the model become phenomenological constants and concentrations. And it can be integrated into the same approach. So we, of course, need kinetic parameters. And if you want to hear more about the latest work about this, please go visit Salah's poster. So is it scalable? So this is an example of variational convenience kinetics, that extension I mentioned to you here. That takes about 240 seconds to solve for the whole body metabolic model. So the problem is quite large. So it's like 120,000 odd constraints and 360,000 variables. So I was working then on the publication for this, and all the time I had kind of seen niggling that at times there was some reactions where the lower axis here is the deviation from kinetic constraints. So typically 10 to the -6 is a good feasibility tolerance, but sometimes there were reactions that were up around 0 or 10 to the -1. So I thought maybe they were lump reactions, or I wasn't really sure what they were. And so I continued to work on integrating like different HMDB concentrations to try and get as many different variables as it could get constrained to reasonable values. And so what I found is, depending on how you choose the value of the C vector, the linear optimization term, you can preference or one constraint over another, right? So or one objective term over another, yeah. So that worked. Then I realized I have a problem actually, because when I started, I said, well, how am I going to explain this? So I started working on the supplementary material, and I was trying to show if I do one constraint at a time and slowly build it up, or one objective term at a time, slowly build it up, how will it look? And then I realized, oh God, it's hard to figure out how to get the right value of the linear objective. So just one objective will definitely outcompete the other, right? Because maximization of entropy is something you do when you have all the rest of your constraints satisfied that you want, right? But reaction kinetics has to be satisfied, right? Whether it's phenomenological or elementary, it has to be satisfied. It's not an option. So I have to get to the boundary for that. But that's competing with, let's say, different terms in the objective. And I had this problem how to achieve that. So essentially what I realized is when you add additional constraints, when you pick your random vector going to the boundary, you might pick one that goes in a direction that's not at the intersection. So previously, let's say picking this direction, that would then take you off down here, and you end up with zero flux. Whereas if I chose a different value of C, I'd be able to go to the boundary that's still within the steady state constraints, for example, but also hit the reaction kinetic constraints. So then I really, that was the scare at bedtime, because I'm like, oh my God, I'm back to Iceland time where years I'd worked on this, and I'm like, because I knew well what the conditions were for convergence, and I thought, oh, is this going to turn out like another monotone or non-monotone situation, and am I back to square one, So fortunately, it turns out not. So one can construct this. So this black area is, let's say, what's eliminated. As soon as the colored area, when you meet with the black area, that's when the constraints are satisfied. So I start off on this random point here, this red point, and one is able to formulate a function that's convex over the nonlinear feasible set, right? So this coloring here, this is a high value of the function, and then this is a highly negative value of the function, and it becomes zero when I hit the boundary. So anywhere on the boundary, this function is zero. It turns out this function is strictly convex, but we need to maximize it. Not normally we're minimizing convex functions, but we need to maximize it to hit any one of these points, and that's possible, and we can do that because the gradient of that is strictly monotone. So how does it kind of look in practice? The algorithm, you have to do a sequence of conic optimization problems, and each time you check what the gradient is of the constraints with respect to where you are, and you converge then via sequence of iterates to the boundary. So this is an example, a numerical example. It's currently just implemented as a first order methods, and so that's why it's not so fast as it could be. I think maybe we could get this down to maybe 10 or 12 iterations, and so you don't actually have to solve the optimization problem completely each time. If you're able to warm start it, maybe you only do. It's much quicker to do. But I was delighted to see it was converging numerically, but I still had this problem. Okay, well, how is it then that if this is converging numerically, I'm not back in the situation I was in ISMB where it's in the end can't prove it. So what turned out was that you have a function, a problem essentially can correspond to a non-monotone variational inequality in the, let's say, the lowest dimension you could represent it in. But that same problem via a transformation to, let's say, optimization over a nonlinear set that's still convex, and via a injective lifting function that lifts the problem up into higher dimensions, can result in a problem then that's a monotone variational quantity. And so this is a really fascinating result, because it's not my result, but it's like a result that's not properly written out in the mathematical literature, right? Because they don't actually know the class of functions that's non-monotone in the lowest dimension, but that can be monotonized by projecting it into a higher dimension. So that's important, right? Because that tells me there's a whole bunch of problems that are considered intractable in lower dimensions that might permit a tractable solution in higher dimensions. So who cares about all this until you can show it's better prediction? So we have a lot of effort done now to underway to try and validate these predictions and other predictions made by complementary constraint-based modelers in the team. And so this is this reconstruction and computational modeling for inherited metabolic disease project. It's a large project involving multiple different omics disciplines and 25 different sites that are recruiting 2,500 patients, and they will get a combination of metabolomics, proteomics, and metabolomics, proteomics, and genomic analysis done. So the idea there is to twofold. Sort of 70% of the project is to improve the diagnostic yield for people with rare inherited metabolic diseases, because maybe at the moment genomics alone can only diagnose maybe 50-60%, depends on the inherited metabolic disease. So we feel, our hypothesis, working hypothesis, by a combination of omic analysis and mechanistic modeling and neural networks and machine learning, we can do better than that. So it's called Recon for IMD because Recon 4 hopefully will be the outcome of one of the outcomes of the project. And so there's a large effort underway by Inez Thiele, Marco Pagni, and Alan Bridge, and their teams, together with some members of my own group, who are working on improving the reconstruction of human metabolism, and especially for lipid metabolism. So there's many other aspects to this project, but one I'd like to highlight to you is work that's ongoing, where we're trying to develop tools that cross-map between different databases, independent of whether you have the correct naming. And so this is done by looking at the structure of the molecule, or comparing the structure of the molecule. So we can talk to Jack about this more at his poster. With such a large, we were trying to recruit from about 120 different inherited metabolic diseases, and with so many different clinical sites and 7 or 8 different metabolomics platforms, or providers of metabolomic platforms, with about maybe 20 different metabolomic platforms, it becomes a very difficult problem to try and organize how you're going to run this clinical study network, right? And so I thought, let's just apply optimization to it, not just, but so Farid fortunately took on this challenge here, and so we wanted to be able to use optimization to identify which inherited metabolic diseases first we should recruit from. So, like, do they exist in out there? Can we model them even at the moment? Can we measure the known biomarkers? How many patients are out there? Which hospitals should exactly recruit which patients, and then which labs should that metabolomic those samples go to? So the results of this is that this optimization approach consistently yields higher let's say, higher, I say, cost effectiveness really overall. But this remains to be seen now. We have to demonstrate that these predictions hold true. So we have recruitment strategy lined out, and many sites are in the process of getting ethical improvement. We already started recruiting. I think we maybe have about 100 patients or so by now. So I'd like to issue now an invitation to those of you who develop constraint-based modeling tools. So some of you may know that I'm heavily involved in development of constraint-based modeling toolbox. It's mostly encoded in MATLAB, but since then there has been different tools in different languages produced, and I think that that's just wonderful. But I wish they had maybe the people, developers, or those that would talk to me about it and say, we're going to recode it, what you're doing in a different language. What would you do differently if you would start from scratch? And if any of them had asked me that, I would have told them several things that I think are wrong with the Cobra toolbox fundamentally. So I didn't start it. It was Marcus Hergard and UCSD. But at the time, it was hard to foresee how things would have gone. But then you get locked in with a certain architecture, right? So for a couple of years, I've been thinking about how we go about the next generation of constraint-based modeling tools. And so this is the current architecture now. We've started to implement it. And if you're interested to get involved in this project, we're hoping to publish the platform in the follow-up Nature protocol to the Cobra version 3 that we submitted, or that's been well cited already. So the idea here is to fix two big problems. First one is the constraint-based modeling tools, most of them out there, they have to run on the same machine as you're operating on your desktop. There are some ones, let's say now, or certainly the Cobra toolbox does need to. There are some ones that don't have any industrial solver dependencies and will run on the HPC machine, but most of them won't. So we want to be able to work with a piece of software that runs on, you can control from your desktop, but runs on a remote machine, whether that's an HPC environment or a GDPR controlled clinical environment, or whatever the case may be. So we also want to be able to amalgamate constraint-based and kinetic and modeling methods developed by multiple different users, independent of the language in which they develop in. Because what's happening at the moment is that the development community is being split. And so Apache Arrow is a data format, interoperable data format developed by the developer of Pandas. And the idea there is that your core data structure is interoperable between languages, and so then we could use multiple methods from different things as long as they input and output and return data in an arrow format. So yeah, with that, I would like to thank my own research group, Inez Thiele, Thomas Hankemeyer, Michael Saunders, and Bernard Paulson for helping sustain this project over the many years that I mentioned. And also I'd like to acknowledge the Recon for IMD consortium, who are doing a lot of effort to gather these clinical data, to analyze the thing, build a new Recon, and many other aspects. So thank you.
Thank you very much, Ronan, for this very enlightening and very detailed talk. Any questions from the audience, please? Maybe I can start with one of them. When you mentioned the multi-site center for collecting the research of the different multi-omics data, do you actually have a way to standardize the way in which the data are produced? Because maybe in different centers they might run different pipelines, or even on the same data set.
Yeah, so whole exome sequencing is by now the standard of care for most rare diseases. So we're intending to do 60 whole genome sequences. And so there's a bit of work to do there in terms of, aligning those to each other. The proteomic data is being generated all by Holger Prokish in Munich, who has a really great quantitative platform. So there's just one provider there. Metabolomics is the big one. So there's a lot of like say 20 different platforms. And the current strategy idea is to do 2 global metabolomic platforms in every blood sample, and then possibly 1 targeted platform. And some of them will already come with some target analysis done in hospitals. And so, yeah, we have a bit of figuring out there to do. It's an open problem. Yeah.
Okay, thank you. Please.
Thank you for the talk. It was very interesting concepts and many things I have to elaborate. So I don't have any questions, but I suggest or comment on your last slides about running the tool, on a server without running simulation on a desktop. And I was wondering whether you consider to exploit the Galaxy platform. I don't know if you are familiar with the LTV network. So it's nice because you have interoperability with other tools. So at the moment, my group is developing, delivering to the community our tools on the Galaxy platform. And so you have a web interface and you just interface with Python or MATLAB or any code you run. So I was just wondering whether you consider the Galaxy platform to release your tools.
Yeah, so the idea here, so I've put in, let's say, virtual metabolic human, and IMD Hub is the let's say clinical study network platform, the GDPR compliant environment, and so but you could connect in any other sort of web interface with this, and so the idea would be that we should have a fairly standardized version of what constitutes a model in here in the middle. Right, and so that should be something like, the standard model structure in MATLAB and bred with the standard model structure in Cobra Pi, bred with SPML. Yeah, so that kind of, it won't be in a flat file format. It's in an in-memory format. But I think if we can manage that, that's the core. Yeah, so but then let's say if Galaxy accepts and let's say initiates and accepts computations in that standardized format, then it should be well possible to integrate with it. Yeah, so it would be. This is not meant to be exclusive here. I'm explicitly opening this project up to people now because it's in development, as opposed to sharing with people when it's done and then people feel it's baked in and where's my flexibility? Yeah, so maybe we can talk more coffee.
Okay, thank you.
Thank you for this wonderful talk, Ronan. I'm very curious about the upcoming Recon 4 and new versions of the human reconstruction. So please allow me a question in which direction the development might go. So I'm wondering, for instance, challenges with tissue-specific models or also gender-specific models, the incorporation of protein structures and such things. Could you maybe say a few words about what we can expect, what Recon 4 could bring to us?
I think that the majority of work is currently on lipid metabolism. So we years ago we had thought lipid maps was going to solve that problem, and it didn't. Partially maybe, but it really didn't. But in fairness, lipid metabolism is so huge. So we teamed up with Alan Bridge, who had been working on this combinatorial generation of lipid molecules. So they're now generating combinatorial. They have generated combinatorially lipid reactions. And so Anastasia is here. You can raise your hand and talk to her about it at her poster later. And so they're certainly we're hoping for a big push now on lipid metabolism. We hope to get every lumped reaction out of recon. Yeah, because for example with thermodynamics they make such a pain because the delta G is so huge, or this sort of problem. Every metabolite should be structured and cross references. No non-structures in later. So that's really what I would love. But what else can you hope for? At the backbone of this work that Jack is doing is a ReconX knowledge graph, and that's designed to be a non-competitive space that people can cross map between different metabolic resources. So it's not designed to have a web interface, because we don't want to make it compete with other resources, but it's designed to cross-link between different things. So Marco is leading development of that, and so there'll be like say a Sparkle endpoint possibly, I guess, for that once it goes public. So if some people are really interested in reconstruction, and you're willing to spend some time on it, months, you contact Alan or Inez or Marco and or myself. But we're not interested in somebody who wants to reconstruct 5 reactions and expects to get on the paper. You have to be willing to commit a lot of time or commit a lot of new data that's not published. So yeah. So you can get out as much as you put in. Yeah.
Other questions? Again, just out of curiosity, as you might know, there are a few people that are very much trying to push the introduction of the inclusion of thermodynamics into FDN. So one of them is Hans Wessel, for example. But other people in the community, let's say, don't include or maybe don't see how to include the thermodynamics in there. Do you think that there's a major advantage of including thermodynamics compared to the other methods that don't do that?
Yeah, so if you look here, where am I with the convenience kinetics? This one. So Wolfram Liebermeister has shown that parameter balance is an essential feature of kinetic modeling. So where are you going to get the kinetic parameters for that? So there's Jack, remind me the name of that community now. That's gathering all the thermodynamic data together. Anyway, I can't remember. I'm very sorry. Elad Noor and a couple other folk are involved in it. I'm sorry if you're here, you're listening. But it's a great effort to basically to catalog all the experimental data done on thermodynamics in one place. So the same thing should be done for kinetic data, because we all try to develop techniques off the same set of data. So why duplicate the effort of gathering the data into a consistent format? Yeah. So open TECR, open thermodynamic enzyme catalyzed reactions. Yeah. Open. Yeah.
Thank you very much. So if you don't have any other, we have questions.
Yeah, I'm sorry, that's maybe a fairly ignorant question coming from a student's perspective, but you've outlined a very involved historical process also in deriving your current approaches, right? I'm not necessarily aware of current educational tracks and materials that would be like the last book I'm aware of that is dedicated to an educational approach to kinetic modeling is by Paulson himself, right? And that's almost 20 years old or something. So I just wanted to ask whether or not you are aware of endeavors in that regard or something that you point to.
Yeah, so I wrote a proposal at the end of last year for a doctoral network. And a heavy component of any of those doctoral and Marie Curie doctoral networks is education. And we were planning to write a textbook in it. Unfortunately, it scored poorly. So we'll probably go back again. So my idea at the moment is, so some of your PIs are interested, is maybe put a Eurocobra consortium together. So get everybody who's really seriously open source software development and maintenance, and get those people together into consortium for Marie Curie. So that's what I'm sort of thinking of now, because you can't write a textbook alone. It has to be representative of multiple people's perspectives somehow, So I think that would be the route. Yeah.
Okay, let's thank our speaker again. Thank you very much.